In creating our model, clays, plastic wraps, colored papers, and sticks are used as the main materials. The clay ball represents the substances moving into and out of the cell, while the plastic wrap functions as the vesicle that either wraps around or fuses with these materials. The yellow paper in the middle serves as the cell membrane where the transport of materials occurs. Lastly, the stick serves as labels to clearly identify each part for easier understanding.
In endocytosis, the orange stick represents the materials entering the cell. As for the pink stick, it shows how the membrane wraps around the materials to form a vesicle, while the clear plastic wrap represents the vesicle itself. The blue stick represents the materials that have already entered the cell through the vesicle. In exocytosis, the orange stick represents the materials that are still inside the cell. The pink stick represents the vesicle containing those materials that is now fusing with the yellow cell membrane. Moreover, the clear plastic wrap represents the vesicle itself. As for the purple stick, it shows the materials that have already been released outside the cell, while the yellow stick serves as the cell membrane. Now, moving on to the presentation.
Starting with endocytosis, it is the taking in or engulfing of materials into the cell by wrapping it with a part of plasma membrane and forming a membrane-bound sac called the vesicle. There are three types of endocytosis, namely phagocytosis, pilocytosis, and receptor-mediated endocytosis. Firstly, in phagocytosis, the cell surrounds a solid material and engulfs it, forming a vesicle, which is then the cell can digest or destroy the engulfed solid materials, such as food and bacteria. And the vesicle that will be formed is called a phagosome. Secondly, in pilocytosis, the cell surface folds back or inwards in attempt to trap the liquid substances that occupies the space outside of the cell before closing in and forming a small vesicle called a pinosome. Lastly, in receptor-mediated endocytosis, the cell surface consists of receptors that identifies the materials around the cell and only picks up or chooses what it needs. When the right material sticks or attaches on the cell surface, it folds inwards and engulfs the materials, thus forming another vesicle.
Exocytosis, on the other hand, however, is the opposite of endocytosis. In exocytosis, it is the releasing of the materials outside of the cell that were engulfed by fusing the vesicle with the plasma membrane. Its process consists of four parts, namely vesicle transport, vesicle fusion, release, and membrane recycling. Starting with vesicle transport, as the word suggests, transport, inside the cells, the vesicle filled with materials move towards the plasma membrane, followed by vesicle fusion, where it fuses with the plasma membrane, which then the release of materials that are no longer needed outside of the plasma membrane occur. After the release, membrane recycling occurs where the vesicle used will be either be fused and become part of the plasma membrane or will be reused for the next transport.
In endocytosis, the macromolecules, molecules that are large enough to fail to pass through normal means, such as diffusion, is engulfed. Once inside, the macromolecules will be broken down into smaller parts, or digested with the help of lysosome, for the necessary needs of the cell, such as maintaining function, providing nutrients, and other usable functions. Moreover, in exocytosis, the broken down macromolecules will then be released outside of plasma membrane, where the macromolecules perform specific functions such as maintaining balance in what the body needs, such as proteins, transportation of hormonal information, and more.
Endocytosis and exocytosis are both crucial processes in life, as they allow cells to maintain balance, eat, and stay healthy. The absence of these processes will result to our structural instability. Therefore, our body won't be able to function properly. Endocytosis helps the cells in providing nutrition and energy to carry on specific functions. Additionally, this process is also useful for maintaining the health of our body, as our white blood cells uses these processes, or phagocytosis, to engulf and digest harmful bacteria that may enter. Moreover, in exocytosis, this process allows the cells to communicate when the macromolecules are released for their own purpose. Though, releasing does not only mean communication. This process also helps the cell maintain cleanliness by sending out damaged or harmful materials out of the cell that possess risk when built up inside, thus maintaining balance as well.
In summary, endocytosis is the engulfing of materials which has three parts, namely phagocytosis, pinocytosis, and receptor-mediated endocytosis. While for the exocytosis, it is the releasing of materials, which has four parts, which are vesicle transport, vesicle fusion, release, and membrane recycling. The presence of endocytosis and exocytosis are crucial to life, as it provides stability, balance, and healthiness to our body. Without these processes, the balance will tilt and we won't be able to function properly.